Today we'll talk about chromosomal mutations. In a way, all mutations are chromosomal because in a way, all mutations happen in chromosomes. But what we mean by chromosomal mutations are those much larger chunks of DNA that are DNA sequence that are affected by a mutation. But we'll get there. So before we do, super brief recap. If you remember, we tend to classify mutations in these two very broad categories. The small invisible mutations, those are the point mutations that change the identity of a nucleotide in DNA, or small indels for insertions or deletions that may change a very small number of nucleotides in a DNA sequence. And we discussed that when those happen, let me take a step back. When those happen anywhere else other than in protein coding sequence, chances are that not much is going to happen. That's the truth. There is a lot of sequence in DNA that is very resistant to these small point mutations or small indels because the information coded in DNA is coded a little more sparsely outside of protein coding sequence. These small mutations tend to have a much more, as we would say in jargon, deleterious effect or harmful effect when they happen in protein sequence, in codons, right? And we discussed these different types of point mutations and the names we give them based on what they do to the sequence of amino acids encoded in those coding regions. So if we were to have this wild-type sequence shown here, a synonymous mutation is any point mutation that does not change the identity of the coded amino acid. Even though the codon has changed, and technically we do have a mutation in there, in that DNA sequence changed, the identity of the amino acid didn't change. Then there is missense mutations, those that do change the identity of the amino acid. And we didn't go into too much detail into that, but there tends to be a sort of a rule of thumb that if a point mutation affects the third base of any codon, there is a pretty good chance that it's going to be a synonymous mutation. And that is because when you look at the genetic codices, those tables that link codon sequences to amino acid identities, you'll notice that for most amino acids, over half of them, there's actually 4 different codons that code for the same amino acid, right? That has to do with the adaptive value of having that wobble position in the third nucleotide of a codon, meaning that the first 2 nucleotides in a codon matter, and then the fourth one, in many cases, it could be any of the 4 available nucleotides, A, G, C, or U, right? And the identity of the coded amino acid is still the same. So when a point mutation happens in the third codon of a base, there's a really good chance, with a few exceptions, that it's going to be a synonymous mutation. Missense mutations, on the other hand, are those that typically target the first or second nucleotide of a codon, because then chances are that it's complete opposite. Almost certainly, you're going to change the identity of the amino acid. And we discussed that the effect of that change depends on where that change in amino acid happened within the structure of the protein and what change took place. Is it a polar amino acid for another polar with the same polarity? Is it a charged amino acid for a nonpolar one, a neutral or worse nonpolar? So depending on the nature of the change and the position of the change, what the effect on that protein function would be. And then we talked about nonsense mutations, those that through a point mutation change the identity of a codon from a coding triplet to a stop codon. And those will create premature termination of proteins. Once again, if this happens near the natural end of a protein, the effect on that protein's function may be negligible. But if it happens near the beginning of a protein, then there's an exceedingly high chance that that's going to be a pretty harmful mutation for that particular gene. That was a brief recap of what we talked about on Tuesday. And then we also talked about these pretty disastrous small indels that happen in non-multiples of 3 that will inevitably change to lead to what we call a frame shift a reading frame shift or frame shift mutations, meaning that the entire message coded by those triplets changes because now those letters get moved one way or the other, and ribosome doesn't know, so it keeps reading in groups of 3, and now the identity of each of those triplets is different, and now you have proteins that have completely different sequences of amino acids, and in many cases you will lead these frame shifts will lead to premature stop codons as well. So these mutations, once again, if they happen near the end of a protein, perhaps the protein is spared or its function is spared, but if they happen anywhere near the beginning or in the middle of a protein, there's a good chance that the protein is gone. So all of that is what we discussed on Tuesday, and all of that is small mutations in coding sequence. And I mentioned I keep saying Tuesday, today's Tuesday. So I mentioned last Thursday that I was gonna briefly go over these small mutations outside of coding region today. So these are mutations that happen before the AUG or after the stop codon. And they can be anywhere in the structure of a gene. So it could be anywhere between the TSS and the site of either termination in prokaryotes or polyadenylation in eukaryotes, but outside of coding sequence, meaning that they would fall in either the 5' untranslated region or the 3' untranslated region. These are fragments of the mRNA that do not code for any amino acids in the protein encoded in these mRNA molecules, but that still carry some regulatory information. So we are going to discuss all of these in much more detail starting Thursday. But when it comes to 3' UTR, for instance, the 3' UTR often harbors sequences that cells use to regulate the stability of certain mRNAs. Cells have evolved ways to actively inhibit the expression of certain mRNAs or even induce their degradation willingly. So these are things that cells want to do, and they do that through sequences they find in the 3' UTR of many mRNAs. And in the 5' UTR region of genes, there's usually sequences that control the translation of the mRNA. Before the cell puts in any energy and time and resources into translating a protein, there are certain sequences in 5' UTR that will slow down the process, or the opposite, somehow accelerate it. So when mutations alter key sequences within the 5' or UTR sequences, those can alter the post-transcriptional regulation of the abundance or location or translation of a given mRNA. I started with the RNA, but point mutations can also happen in the promoter sequences of genes. So these are mutations that happen in DNA, like all mutations, but don't affect the sequence of nucleotides in the mRNA because they're never transcribed, right? The promoter is not transcribed. Anything that happens upstream of the TSS. But those sequences can still have a pretty important effect on the expression of those genes because they can affect regulatory elements in the regulatory promoters, or they could wipe out important consensus sequences in the core promoter. So you may have a sequence that fell outside of the transcribed sequence in a gene, and yet turn off the expression of that gene altogether if those mutations alter some of the consensus sequences that are critical for those core promoters. So in core promoters, they tend to be pretty harmful. In regulatory promoters, they can go either way. They can either inhibit somewhat the expression of a gene, they can enhance the expression of a gene, or they could, in some cases, even alter the expression patterns of a gene. Based on what we were discussing last week, that those type of mutations would get into the realm of those famous neomorphs, when a gene all of a sudden is not doing anything more potently or less potently, but it's doing something new, okay? For instance, a gene that starts being expressed in cells that normally isn't expressed, or at times when cells normally wouldn't express a gene, and now these mutations will cause them to be expressed. for regulatory promoters. So it can inhibit their expression, enhance their expression, make it higher, make it lower, or change the pattern of expression. Regulatory promoters are the sequences. We'll discuss all of that in more detail starting Thursday, but regulatory promoters is where the logic for expression of a given gene is written in DNA. So those are the collections of sequences that cells use to determine if a given gene that they should express or not. And mutations in those sequences could alter the logic of expression. So a cell that normally doesn't express a given gene now all of a sudden decides that it should express it, or the other way around, and so forth. Yeah? Then we have here a circle around the terminator or the poly-A signal. Now, by definition, these mutations, let's say that a mutation takes out that very specific poly-A signal sequence, the AAUAAA sequence that is the poly-A signal in a eukaryotic gene, or takes out a terminator signal in prokaryotes. That will not affect the sequence of amino acids of the protein because by definition, these things are after the stop codon, right? They're towards the end of the gene. But the way that these mutations can have a large effect on the expression of these genes is by now generating longer 3' UTRs. And as I was saying before, and as we'll discuss in a bit more detail next week, 3' UTRs is where much of the post-transcriptional regulation of certain mRNAs takes place at a biochemical level. So by extending the 3' UTR region of an mRNA, you might be adding functions to it, or you might be adding certain regulatory logic. Let me throw out a quick example. Let's say that by extending, a mutation takes out the normal poly-A signal of a eukaryotic gene. So now that mRNA will be a little longer than usual because the polyadenylation machinery will not see that poly-A signal. It may find one later, may find one downstream, but who knows how further downstream, and now there's more 3' UTR in that gene. It could be that added 3' UTR, and this was not meant by evolution, but it could be that now that this extra 3' UTR has some of these sequences that allow cells to attack and inhibit the expression of certain mRNAs. So now this mRNA that should not have been inhibited, whose expression should not have been inhibited, is now being inhibited because of these extra signals in its 3' UTR. So even though all of this happens outside of coding region and will not affect the identity of the amino acids being stitched together in that protein, it still affects the expression of those genes simply by adding extra sequence to the 3' UTR. Is there a question there or no? Core promoter, if you remember, the core promoter is that sequence where the transcriptional machinery assembles. So in bacteria, it is the -10 and -35. The sigma factor binds, and RNA polymerase comes in and starts transcription. In eukaryotes, it's any collection of either TATA boxes or GC-rich boxes or CAT boxes, any combination of these consensus sequences that will lead to the assembly of the TF2 family of proteins. Once a bunch of these TF2 proteins assemble at the core promoter, they bring in RNA polymerase and transcription begins. Now, in contrast, there is the regulatory promoters. These are sequences that will accelerate or inhibit the formation of this transcriptional machinery. So on the one hand, there is the core promoter that genes need to assemble the transcriptional machinery, and then there's regulatory promoters that will speed up or slow down that process. Okay? All right, and here, last, not shown in this diagram, there could be mutations in DNA sequence that affect splicing donor or acceptor or branch points, consensus sequences that can affect the final composition of a eukaryotic mature mRNA if they affected the way that those pre-mRNA molecules are spliced. The reason I say that it's not shown here is because here we're going from DNA to a mature mRNA and we're not showing the intermediate that is the RNA with all of those introns that need to be spliced out. But should any mutations occur in those splicing donor or acceptor sites, those will affect directly the way that a pre-mRNA is being spliced and how exons are stitched together, and that could affect the final composition of the mRNA. Okay? So the fact that these small mutations happen outside of protein coding region doesn't mean that they won't affect the expression of the gene. They will not affect the amino acidic sequence of that protein, but they can certainly affect the expression and therefore function of that gene. Right? Sometimes in harmful ways, sometimes in beneficial ways. Sometimes they create that playground for evolution to explore new functions for a gene in a given environment, and it may be the basis for new adaptations and new abilities for organisms. So now we can move on to those larger visible mutations. And when I say visible, I mean that these are things that you can see with a regular microscope, as long as you know what you're doing and you know how to carry out certain techniques for seeing chromosomes inside of cells. And these larger visible mutations are divided in aneuploidies and chromosomal aberrations, and we're going to discuss those next. So first, let's talk about aneuploidies. It turns out, so chromosomes, for the most of the lifetime of a cell, when the cell is not actively dividing, they exist as one might imagine them. We're going to discuss this in more detail starting Thursday, but you might imagine chromosomes existing as like long spaghettis that are sort of in a pretty, not chaotic, but a pretty unstructured conformation within the nucleus. Yet, when I say chromosomes, the immediate mental image that may come to mind is those famous X-shaped structures that we associate with the word chromosome. So both are real. Chromosomes are simply DNA molecules. It's just that the famous X-shaped structures occur when cells are about to divide and they need to divvy up the DNA between the two daughter cells. And it's kind of hard to divvy up DNA between two daughter cells when these are long strings that can get all tangled up. So prior to division, cells will condense those very long DNA molecules into these much super highly condensed structures and adopt these more familiar X shapes, right? So what for many years now, decades now, we've known how to do is to stop the process of cell division at a stage where we can easily, using a regular light microscope, see these chromosomes. And that's why we call them visible. You still need a microscope, but you don't need anything fancier than that, right? And that gave rise for a long time to things like this. For decades, people used these things, just looking at these structures under the microscope. And these allow people to determine the number and the types of chromosomes that were present in cells in an individual. And the collection of all chromosomes in an individual is what we call the karyotype. Okay? It's the karyotype. It's the collection of organization of chromosomes by homologous pairs. It's the collection of chromosomes that exist in an individual. More recently, things got technically a little fancier, and what people will do is a technique called fluorescent in situ hybridization, or FISH for short, by which you take the same chromosomes that normally you would see them as dark structures against a bright background and incubate them with different molecular probes that will recognize different sequences that we know are associated to different regions of the different chromosomes in human cells. And each of those probes will fluoresce in a given color, in a given wavelength, right? So you end up generating these pretty beautiful images of the different chromosomes with different colors that are associated with their different regions, right? So on the left, we have a normal human karyotype. Whoops, I didn't mean to. Whoa. On the left, we have the normal human karyotype, and you may have heard this before. The normal human karyotype is 46 chromosomes. It's 23 pairs, and those 23 pairs divvy up between 22 somatic pairs and 1 sex pair, right? So in this case, we go down the list, and there is 1 through 22 for the somatic, and then the pair of chromosomes, which in this particular case happens to be XX. Okay? So the normal would be, it's 22 somatic plus a sex pair. If you take a look at the karyotype on the right, what's wrong with it? I'm missing 14 and an exon 5. Correct. I kind of doctored this image, but this is not much different from what someone interested in finding out the karyotype of an individual would do or see, right? The presence or absence of certain chromosomes, based not only on their size, but also those combination of colors that you expect from them. I didn't do it here, but this technique allows you to not only see the composition by in terms of the number of chromosomes and their identity, but also make sure that the distribution of colors is quote unquote normal. So if we zoom in on chromosome 10 here, we see that both of them have a purple part in the centromere and much of both arms, and then these green part or these green probes attaching to them near one of their telomeres, right? Fish might allow us to see changes to this, might allow us to see how one of the chromosome number 10 is all green, for instance, or all purple, right? Both of which would indicate that there's something wrong with those chromosomes. So these fish karyotypes allow us not only to see gross changes to the number and identity of chromosomes in a karyotype, but also more subtle changes that have to do with the actual molecular composition. So, aneuploidy is the name that we give to any number of chromosomes in cells that is abnormal. So, euploidy is the normal number of chromosomes in cells for a given species, and euploidy is no normal, right? So, abnormal number of chromosomes. And the reason why a cell may end up with an abnormal number of chromosomes in it is a phenomenon called non-disjunction. So during cellular division, and in this case we're first addressing mitosis, if you guys remember, in mitosis, in this particular cell, and for cartoonish purposes, we're looking only at two pairs, or two chromosomes, rather. So this particular cell has only two chromosomes, and during one of the latter phases of mitosis, the two sister chromatids that were created following DNA replication of both of these chromosomes will separate and migrate towards the opposite ends of that dividing cell, right? So during anaphase of mitosis, they will migrate towards opposite ends. And when the cell finally divides, both the daughter cells will have that normal number of chromosomes, those two chromosomes that the mother cell had as well. So during normal mitosis, the sister chromatids separate, and each of the sister chromatids migrates to a different pole of the cell and will end up in one of the daughter cells. Now, this is what we call chromosome disjunction. Disjunction is sort of a fancy term to say separation, right? So it's chromosome disjunction. This is what happens normally. When there's chromosome non-disjunction, that separation of the sister chromatids fails. For one or more of the chromosomes, it just fails. So instead, when we look at these black chromosome down here, instead of the two chromatids separating from one another, migrating to opposite poles of the cell, they both migrate together to one of the poles of that dividing cell, and they will end up together in one of the daughter cells and missing from the other. So one of the daughter cells ended up with a number of chromosomes that equals 2n plus 1, and the other one will be missing a chromosome, so it's a 2n minus 1. Why is this a problem? It is a problem because even though there's been no mutation to the sequence of nucleotides in DNA, there's been no genetic mutation in that strict sense that we discussed last week, that there has to be a change in DNA sequence. Sequence of DNA hasn't changed here. It's just that one of the daughter cells ended up with an extra copy of a chromosome, and the other cell is missing one. So for the cell missing the chromosome, it's kind of easy to understand how that will be a problem. There's a whole chunk of genes that are not there, right? And then for the cell that ended up with an extra copy, it can also be a problem because of something we call genetic imbalance. So when you lose genes, it's obviously a problem. You don't have them. Normally, you would have them for a reason because you need them, but now you don't. So that is clearly a problem. But sometimes when you end up with two copies of the same chromosome, you may have too much of those genes being expressed, and that can be a problem as well. Yes?
Can this process also happen during meiosis?
Meiosis, yeah, and we'll discuss it next. Yeah, this process can also happen during meiosis. So just hold on to that idea of the genetic imbalance. If this chromosome non-disjunction happens very early on in the sort of the developmental history of an individual, let's say it happens in the first division of the zygote. Right after fertilization, the first cell that will make up an individual undergoes a chromosomal non-disjunction in its first division. Now, the first two cells that will form up that organism are already aneuploid. One is missing a chromosome, the other one has an extra one. Let's say that from that point onwards, everything is just fine. Everything's just fine from that point onward. No more chromosomal non-disjunctions. All of these new cells don't know any better. They will copy all of the chromosomes they have, make two chromatids out of them, divide them up during mitosis, and continue with life as normal, thinking that for half of the organism, they should have 3 chromosomes. For the other half of the organism, they should have 1 chromosome. Right? Obviously, these can be pretty disastrous. So some organisms tolerate it a little better than others. In mammals, it's disastrous. It would quickly lead to something we call embryonic lethality, with a few exceptions. In other organisms, it can be tolerated. So here in this picture, what you see is a fly that half of it, I don't know if you can appreciate it, but half of it, if we can draw a line right in the middle of this fly, and one half has clear eyes, whereas the other one has red eyes. The body coloration on the left half of this fly is lighter than on the right, and I know that you can't appreciate it because of the wing here, but trust me, this fly, believe it or not, half of it is male, the other one is female, right? And this happens more often than you would imagine in simple organisms like fruit flies because of chromosomal non-disjunctions that happen very early on and basically made the organism half of it contains 2 X's and the other half contains only a Y. So one half of the organism thinks that it's female and the other half thinks that it's male, and some of these body coloration differences and the eye color are associated with the presence or absence of these chromosomes. Okay? Now, those are kind of exceptions. More often than not, across species, chromosomal non-disjunctions that happen early in development end up being pretty disastrous and lethal. What if these chromosomal non-disjunctions happen in an adult organism with millions of cells in it? You can think a human being or any larger animal or plant, right? So my question to you is, we just discussed that aneuploidies can be fatal in early embryos. Can aneuploidies have any effects on fully formed adults, where there will be some cells undergoing division once in a while, and any of those could be subject to a chromosomal non-disjunction? Would they have any effects on the organism as a whole? Klinefelter. Did those happen in adults, or do they happen from early embryogenesis?
I think it's from early embryo.
The question was about fully formed adults.
can't there be like skin discoloration? Because I know sometimes like if you're exposed to too much sun, your skin can be like permanently discolored.
Discolored.
That could be caused from chromosomal changes.
Okay, but we're making a big assumption here that it was through a chromosomal non-disjunction. I do understand what you're saying, what you're describing, that exposure to radiation could discolor certain patches in an irreversible and permanent way, but I don't know if it's through disjunction, right? Or non-disjunction.
No, I was just going to say, wouldn't that be more of like UV damage to the cell?
And that UV could be causing permanent damage in the form of genetic mutations, not necessarily these aneuploidies. Yeah.
Can you take like cancer as an example?
Okay.
And like, wouldn't that still, you can get cancer later on in life, and like that would still be an example.
But how do we link an aneuploidy to cancer? I like where you're going, but how do you make that connection?
So I was gonna say, going back to Down syndrome, since it's trisomy 21, I believe it's older women that are higher risk with getting Down syndrome, as well as trisomy 5.
Oh, I see where you're going. So what you're saying is that what leads to, going back to this idea of non-disjunction in meiosis, what leads to, in the next generation, the increased probability of Down syndrome or trisomy of 21, it could be a consequence of non-disjunction in a fully formed adult. I accept what you're saying, I like where you're going, but that's an effect that will happen in the next generation. That older woman is not necessarily affected by that non-disjunction. It's not like she suffers any of the consequences of that trisomy of 21.
What about if it was trisomy 5, because females leading with high risk of miscarriages?
Again, that's something that I think you're thinking what affects her children.
Okay.
If it was in a high division area, maybe like in your stomach lining,
Uh-huh.
something like that,
Uh-huh.
and it in some way was specifically had genes in it that helped with like the function of the stomach lining, and it then reproduced.
Okay. Okay. So I'm gonna reiterate for everybody. So the argument here is, when you look at a diagram like this and you see that the aneuploidies are affecting 1 out of, 8 or 10 cells, if an aneuploid is going to be affecting, best case scenario, 1 out of 10, 10%, worst case scenario, 1 in a million, who cares, right? But her argument is, well, but if it's an organ that has a high turnover, right? Those cells that are aneuploid might perhaps accumulate, and they may contribute through their misfunction to a misfunction of the organ, right? Cool, I like that. So depending, you have some sort of chromosomal aneuploidy in an organ that has trillions of cells, and it happened in one cell, maybe you don't notice it. You have the same thing go on in, among neural stem cells in the brain or smaller organs like the pancreas, and you might start noticing the effects. I want to go back to that. So, have you thought about it?
Okay, yeah. Like, what if you took melanoma, for example? Like, someone is so exposed to UV rays constantly.
Yeah.
And what if that just damages the cells, like causing like cell death or to just be like dysfunctional? And if there were to be any more like dividing or whatever you said, it wouldn't be able to. So would it also have an effect or play a role in that?
So we keep going back to this idea that UV is damaging the DNA, right? In this example, there's something that goes along those lines. So let me sort of reveal, because that's more or less where I was going with this. Without any damage to the DNA sequence, an aneuploidy can, for all intents and purposes, serve as if there had been a mutation. Why? Let's think of that cell that is now missing a chromosome. If that chromosome contains genes that were important for not letting that cell divide uncontrollably, without those genes, now this cell has nothing to stop it. And it will continue dividing. It doesn't know any better. The genes that would normally tell you, hey, you shouldn't divide, we're in an adult. We should replace cells once in a while, but you don't need to. You're not in a baby, you don't have to grow, right? If that cell missed those genes through the aneuploidy, now it might start dividing uncontrollably and in the process make more and more mistakes, and in the process accumulate extra genetic mutations that may eventually lead to cancer. The same argument could be made of the cell with the extra chromosome if that extra chromosome happens to have those genes that actually stimulate a cell to divide, right? So let's say, go back to skin. Skin cells divide. We are constantly replenishing skin, right? If one of those divisions were to lead to an aneuploidy and the chromosome that got lost happens to bear these genes that keep the cells in check when it comes to division, let them divide when it's needed, but not otherwise, then there could be a tumor. I don't mean to scare anybody. In fact, there is a much higher chance that you might develop some skin lesions and malignancies through UV exposure and actual damage to genes than through aneuploidies, because what happens is when a cell loses a chromosome, for the most part, there's so many functions that it will lose. Imagine all the genes that we have, and they're divvied up in 23 or 22 somatic chromosomes and a sex pair, right? So there is so many functions that are lost that a cell that misses an entire chromosome will tend to die, right? But it's not completely out of question that an aneuploidy might lead to a phenomenon like this. So even in adult organisms, an aneuploidy that you would think is affecting a cell out of millions could have a pretty harmful effect. Okay? All right. Now let's talk about those meiotic non-disjunctions. So in meiosis, during normal meiosis, there's two divisions. The first division separates the pairs of homologous chromosomes from one another. So in this case, with this cartoonish cell that had only 2 chromosomes, we have to assume that they were the pair of homologous chromosomes in this silly species with 2n equal 2. So the first division will separate the white chromosome from the black chromosome into the 2 daughter cells of meiosis 1, and then the second meiotic division will separate the chromatids, leading to 4 gametes, 2 of which have the chromatid from one homologous chromosome and the other 2 from the other. So there's 2 instances in which there is chromosome disjunction. The first disjunction separates the 2 members of a homologous pair. The second disjunction separates the 2 sister chromatids within a chromosome. Either of those 2 could fail. And if you have a non-disjunction during anaphase 1 and the 2 homologous fail to separate from one another, now both of them will end up in one of the daughter cells, the other one inherits nothing. So now half of your gametes after the second meiosis, half of your gametes will have an extra chromosome and the other half will have chromosomes missing. Right? So for all intents and purposes, a non-disjunction that happens in meiosis 1 affects 100% of the gametes. All the gametes are abnormal. Okay? Sometimes the non-disjunction happens only in the second meiotic division. So the 2 homologous separate in meiosis 1, that worked, but then the disjunction of the sister chromatids in meiosis 2 fails in one of them, and now one half of the gametes are normal, they carry 1 chromosome as they should, but then the other half of the gametes, some cells will have extra chromosomes, some cells will have some missing. So depending on when these non-disjunctions took place during the process of meiosis, what effect they'll have. So here you see 2 individuals. The individual on the left is doing meiosis just fine and all the gametes are normal. The individual on the right is undergoing a non-disjunction of meiosis 2, so half of its gametes are going to be faulty. Now what this cartoon is meant to show here is that even in that case, things could be fine. If it turns out that this individual on the right is passing on one of the gametes that was normal, no one will ever know that there was a meiotic non-disjunction. No one will notice, right? On the other hand, if the gamete that participates in fertilization is either of the ones that was aneuploid, then the zygote that comes from this fertilization event will be aneuploid itself, and depending on the species, most likely won't complete development because there is embryonic lethality associated with gene imbalance. Yes?
Is that how, like, sometimes some babies, when they're born without, like, certain limbs? Is that caused by this?
No, the lack of certain, so we'll talk about human aneuploidies in the next slide, but the lack of a limb has to do more with issues that happen during the development of that fetus. They could have a genetic basis, but it's not as simple as to say one chromosome missing, one limb missing. One other chromosome missing, one organ missing. It's not that straightforward. The genes for making limbs and organs for what we normally call organogenesis are distributed throughout the entire genome. So there's no matching of a given chromosome to a given function, right? With the only exception that we'll discuss in a few weeks when we talk about sex determination. That's right. So the first two cells, the ones that come from meiosis one, I don't know if you can see that from the back, but these two cells here, they're not normal. One of them is aneuploid, right? I mean, sorry, I misspoke. No, you're right. So the first two cells are normal. They both inherited one of the homologous chromosomes. It's just that the non-disjunction happens in one of them during meiosis 2, and that's the one that leads to faulty gametes. Does that make more sense now? Yeah? So under normal circumstances, you would, in these species where the diploid number is 2, you would expect that the gametes have only one chromosome, and the fully mature gametes will have one chromatid for each of one of those chromosomes, right? In non-disjunction in meiosis 2, half of the gametes are normal, but then the other two are aneuploid. One of them has an extra, one of them has one missing. When the non-disjunction happened in meiosis 1, all of them are affected. Okay, so let's talk about those human aneuploidies. There are a few human aneuploidies that are tolerated during embryonic development, and that leads to healthy adults, or relatively healthy adults. So what I mean is that they can get through development. So Down syndrome is trisomy of 21. And the reason why trisomy of 21 is tolerated is 21 is a very small chromosome with fewer genes than many of the larger chromosomes. So the genetic imbalance is there. There is an extra chromosome 21 in individuals with Down syndrome, but the presence of these extra genes it's tolerated by the embryo and later the adult, right? Something similar happens with trisomy of 13 and 18, both relatively small chromosomes. These are tolerated during embryonic development, but these trisomies are generally associated with lethality during the first year of life of those individuals. And then another very small chromosome in humans is the Y chromosome. So individuals who have a trisomy in the sex chromosome such they have the configuration XYY, an extra Y, they can tolerate that extra chromosome because, once again, the Y chromosome carries very few genes, and the extra dose of those genes, it doesn't really affect much. So the embryo tolerates the genetic balance, and these individuals make it to adulthood no problem. They may suffer sterility, though, because when cells in these individuals try to pair up the sex chromosomes, the homologs, the sex chromosomes, during meiosis 1, they can't. They find an X and two Ys, and that confuses the cell, and that may lead to problems during gametogenesis. So these individuals tend to suffer of sterility. Then there's a whole series of aneuploidies associated with imbalances in the number of X chromosomes. Now, X is gigantic. It's one of the larger chromosomes in humans, and it has tons of genes. So how come that an aneuploid of such large chromosome can be tolerated, right? How can all of these individuals that are XXX or X nothing or XXY reach adulthood? The reason for that has to do with something that some of you may have heard before, most of you maybe not, and wouldn't blame you. In humans, females are XX, males are XY. And the presence of that extra X in females, or that missing X in males, depending on how you want to look at it, creates sort of a difference in the genetic load associated with the X chromosome in either sex, right? So if you have an extra X in females, you would imagine that there is too many of those genes or too few of those genes in males. So how did evolution correct for that? Through a process that is called dosage compensation. So what happens in humans and in many other species is in females, one of those X chromosomes is shut down. So even though females have 2 X chromosomes throughout the organism, in each of those cells, one of the X chromosomes is completely shut down. So now effectively, the dosage of genes in the X chromosome in males and females is the same. Because of this shutting down process is that all of these aneuploidies can be well tolerated. So in XXX females, these are females that have an extra X because of an aneuploidy in one of the gametes that led to an extra X in these individuals. Those aneuploidies are tolerated because instead of one of the X chromosomes being shut down, 2 of them are. The mechanism is such that it works in such a way that it leaves one of the chromosomes on. So if you put a 3rd chromosome there, it will shut it down, and it's fine. In Turner syndrome, females that are X and nothing, they have lost an X chromosome. Same thing, because the missing chromosome would have been shut down anyway. An X nothing, for all intents and purposes, is the same as an XX because one of the X's is shut down anyway through dosage compensation. And in Klinefelter syndrome, XXY males, these individuals are males, as we will discuss when we talk about biological sex determination, because they have the Y chromosome, and the Y chromosome has genes that guide the embryo towards male sexual characteristics and dimorphisms. But then there is the 2 X's. One of them will be silenced. One of them will be shut down, as it normally would in XX individuals. So all of these are well tolerated, and these individuals reach adulthood, completely normal lives, except, once again, when it comes to gamete formation. And that's often how these syndromes clinically manifest. Because these individuals, when they're trying to form gametes, those cells have a really hard time pairing up sex chromosomes during meiosis, and meiosis fails, so there is no formation of gametes.
Wait, so for normal humans, for normal female, one X is activated, the other is inactivated.
Yes.
For males, both X, Y are
Yes, so you can say that the X is not inactivated, so it's functioning, and the Y is not inactivated, but again, it has a handful of genes. It doesn't really contribute much.
not too much of
More lethal. So again, for any other chromosome, that rule may apply, but for the X chromosome, it doesn't apply so much because even though a cell may have more than one X chromosome, it will silence one or more anyways. So all cells in humans operate with one functioning X chromosome, as it is. Does that make sense? Yeah. So the triple X and the Turner syndrome, X nothing, at the end of the day are the same. The one X in Turner and one of the X's in triple X are working. The extra 2 in triple X are shut down, and the one missing, the cells won't even notice.
survive
through development.
Doesn't account for the life quality. When they say quality, it means
Yeah. When I mean, so it is tolerated during development. Now, I will also say that these aneuploidies of the eggs don't have any major effects on the life of the individual later on, other than infertility.
So basically, when they're alive, it just means you have the most ideal conditions for them.
I wouldn't go there. So that's passing judgment, right? So if individuals with these aneuploidies value procreation, for instance, then it's a huge issue. But it depends on the moral value that you put on certain things. So I wouldn't say that being infertile is necessarily means a lesser quality of life or anything like that.
Well, couldn't the additional chromosome or missing chromosome also affect other traits?
For the most part, they don't, because there's no real genetic imbalance, because normally the second X gets silenced anyway. So you take any XX individual and they're operating with one X anyway. So having an extra X, the two get silenced, they operate with one. Having a missing X, it's fine. The one that they have operates. All right, let's get through this real quick. We're gonna turn the page and talk about chromosomal aberrations now. And that means when the number of chromosomes is normal, but like if you were to look through a fish, you would see that the composition of those chromosomes has been altered somehow. So we call those chromosomal rearrangements or chromosomal aberrations. How do they happen? So anytime that there is what we call a double strand break in DNA, both strands of DNA break anywhere in the DNA molecule, those breaks can be repaired through two mechanisms. One of them is called non-homologous end joining. That's a mouthful just to say that there's a bunch of proteins that recognize the break, the broken ends of those DNAs, those DNA fragments, and they come together and they restitch them together just because. So non-homologous end joining means that there is end joining of broken ends of DNA molecules that is not based on any sequence that is present in those broken fragments. These NHEJ proteins detect the broken fragments and bind to them, and once these proteins, NHEJ proteins, see each other, they come together, and then they glue the two fragments together. For the most part, if there's been only one double-strand DNA break in a DNA molecule, the only place where this is going to happen is where the break took place, and NHEJ will fix the issue. That's why NHEJ evolved as a mechanism. It's very advantageous. It fixes many of the problems. Now, every once in a while, there is more than one double-strand break. So here we have a cartoonish scenario in which two breaks happen, one between locus A and B, and one between locus B and C. The NHEJ proteins will recognize the broken ends, bind to them, and start looking for one another to do this stitching, nilly-willy, without really paying attention to sequence, right? And if they find each other in the right configuration, it's fine, nothing happened, no mutation. The broken ends were rejoined as they should, and no one notices the problem. But there can be instances in which, let's say, the NHEJ proteins bound to fragment A rejoin with those in fragment C, and the two ends of B rejoin to each other, in which case we're going to end up with this A bound to C configuration and B bound to itself in a DNA loop molecule. I see some faces going like, what? Yeah, exactly. So what happens is this DNA loop, this circular DNA molecule, is recognized as something that doesn't belong in a eukaryotic cell and gets destroyed. But more importantly, the A and C fragments are now joined together with nothing in between them. So we say that a deletion took place. More specifically, a deletion of the B locus took place. Right? We used to have ABC, and now we have AC. No B in between. There may be cases in which, and it's all here in the cartoon, but the A of N joins with an N of B, but the other one, right? So there's this swapping of N's such that now B is rejoined in between A and C, but upside down. So there's a flipping of the B locus, and that leads to what we call an inversion. The B locus has inverted. And we can get creative, but there can be double-strand breaks that happen in different chromosomes, right? And depending on how the end joining goes, there is always room for no mutation to take place if the broken ends happen to rejoin with each other as they were before the breaks. But if there's some swapping of broken ends, there can be entire chunks of a chromosome that are swapped between them. And we call those translocations. So originally we had two chromosomes, one with loci A, B, and C, the other one with loci D and E. And after translocation, you could say that E translocated to A, or that B and C translocated to D, or there was some sort of swapping between these two. Yes?
So then, is there a difference between translocation and transposable?
There is. So translocation is an entire region of a chromosome moving to a new location, whereas transposons, no matter how large they are, they're much smaller than an entire chunk of a chromosome. And those transposable elements are, in a way, they act independently of any of these strand breaks or any faulty repair mechanism. For a transposable element, this jumping around is part of their natural cycle, whereas this is a consequence of something that went wrong twice. The double strand break that led to these fragmentations and the faulty repair. Does that make sense? Yeah? They sound very similar, but a translocation and a transposable element will be different.
So there is no case of replication, right? That's impossible. So replication is impossible in this case.
No, it's absolutely possible, but DNA polymerase doesn't know any better. So when DNA polymerase goes through this new chromosome that has A and E in it, will copy A and E. And when it goes through DBC, it will copy DBC. Remember, there's nothing in DNA polymerase that can detect whether the sequence that it is replicating is normal or has been mutated somehow, including these translocations, deletions, inversions. DNA polymerase doesn't know any better, and it will make new copies of these mutant chromosomes. Okay? All right. It's a race against time here. So quickly, what's the consequence of deletions, inversions, translocations? When you think about it, except for deletions that may take a chunk of sequence out of the genome, in general, all of these, they can lead to genetic imbalance, but sometimes they don't. Why? Because the inversions and the translocations are movements of sequences within the genome, but the sequence is not gone. Now, there are some mechanisms by which inversions and translocations can lead to genetic imbalance, and we'll address them. But more importantly, all of these, and that's why it's important that we remember that we call them chromosomal rearrangements. All of these chromosomal rearrangements lead to issues when cells try to pair up homologous chromosomes during meiosis. Right? During mitosis, each chromosome is its own thing, and cells will treat each chromosome in the cell independently. But during meiosis, the cell needs to pair up the homologous chromosomes, form those homologous pairs, and if there's been translocations or inversions or deletions, the cell may fail to recognize the two homologous chromosomes belong with one another. Right? So if the cell in the previous example was trying to pair up the new chromosome that has AE with the original chromosome that had ABC, it may have a hard time recognizing that these belong together. Right? That is to say that when deletions, inversions, translocations take place in their germline, those germ cells will have a hard time going through normal meiosis and generating gametes. In somatic cells, the story is a little different. So deletions obviously will lead to a genetic imbalance. Many genetic imbalances are tolerated by somatic cells, especially if these deletion affected what we call a haplosufficient gene, meaning that a gene for which half the doses is enough, right? So haplo means a half, and sufficient means that it's enough, right? So haplosufficient genes are those for which half a dose of the gene is fine. So if there's a deletion affecting one of those, cells won't even notice. There may be other genes that are haplo-insufficient, for which half a dose is not enough, and those may suffer consequences. And let's say, back to our example of a gene that normally controls cells and stops them from dividing when they shouldn't, if the deletion takes out one of those, then obviously there can be severe consequences for the organism because those cells will start dividing uncontrollably and lead to the formation of a tumor. Eventually, they could lead to the formation of cancer. With inversions and translocations, they can lead to genetic imbalance if these breakpoints and the points of rearrangement and reorganization affect coding sequence, right? So if, let's say that there was a gene here that started in locus A and went all the way through B, now half the gene has been inverted, so the gene that was coded in locus A is now broken. So these inversions and translocations can have an effect on the expression of genes. And they can also have drastic effects if they involve non-coding regulatory sequence. And a preferred example of this is shown in this very last slide. Is an example of a very common translocation that happens in humans. Right? The reasons for why this translocation is common has to do with the fact that these two areas are particularly sensitive to double-strand DNA breaks, given sequence, given exposure in cells. We don't know exactly why they're so prone to undergoing those breaks. And because they're more prone to undergoing those breaks, they're more prone to suffering this translocation. So when the translocation between chromosomes 9 and 22 take place, there is one chromosome ends up as mostly 9 and a little bit of 22, and the other one is mostly 22 and a little bit of 9. And that one is called the Philadelphia chromosome because this was discovered some lab in Philadelphia. And what happens with that chromosome is that it joins together the regulatory sequence that controls the expression of a gene called BCR with a gene that drives cells to proliferate. Why is that a problem? Because BCR is a gene that is normally expressed in B cells of the immune system. Right? That regulatory sequence is telling B cells, please express the BCR gene, we need it. Please express the BCR gene, we need it. Please express the BCR gene, we need it. But after the translocation, they keep telling the cell, please express this thing. And now the cell is expressing this other gene called ABL that is like an accelerator of cell divisions. So these B cells that underwent the 922 translocation, not only are they not expressing the BCR gene that they needed, but also they can stop dividing. And that leads to the formation of lymphoma, okay? One of the blood cancers. That make sense? This is from a previous slide. So I was simply trying to, everything that I just said relates to a translocation. The B is inverted here because in the previous slide I was showing how deletions or inversions may not affect gene dosage, but they could affect the expression of genes if those breakpoints happen to fall within coding sequence.